So friends, now we are going to talk about the gram staining procedure. So gram staining, it is one of the most important staining procedure in microbiology. It is one of the most important staining procedure in microbiology laboratory that is used. And this gram staining, it was invented by the scientist Christian Gram. It was invented by the scientist Christian Gram in the year 1884, and it is one of the most important staining procedure in microbiology. So this staining technique, it is a type of differential staining technique. Staining technique, it differentiates the bacteria into two types. That staining technique, it will differentiate the bacteria into two types, that is gram positive bacteria and the gram negative bacteria. So the differentiation of the bacteria, it is made into two types, that is gram positive bacteria and gram negative bacteria by the gram staining. And the gram positive bacteria, they will retain the primary stain, they will retain the crystal violet, and they will appear purple in color. The gram negative bacteria, they will lose the crystal violet and they will retain the counter stain. Means they will retain the safranin or basic fuchsin and they will appear red or pink in color. Okay? The cell wall of the gram positive bacteria, it is thick, and the cell wall of the gram negative bacteria, it is thin and it is more complex. The cell wall of the gram positive bacteria, it is thick and it is containing more peptidoglycan. So the content of the peptidoglycan in the cell wall of the gram positive bacteria, it is more, and the lipid content in the gram positive bacteria, it is less. Then into the case of the gram negative bacteria, it is thin and complex. The peptidoglycan content into the gram negative bacteria, it is less, and the lipid content in the gram negative bacteria, it is more. Gram staining, it is based on the cell wall composition of the bacteria. So in the gram staining technique, 4 reagents they are used. In the gram staining, 4 reagents are used. So first reagent, it is primary stain. The primary stain, it is the first staining reagent used in the gram staining, that is primary stain. And the example, it is crystal violet. It is the primary stain. Then the second staining reagent, it is mordant. That is the second staining reagent. So grams iodine, it is the same mordant, second staining reagent. Then decolorizer. So decolorizer, that is 95% ethanol. It is used as a decolorizing agent. And fourth one is the counter stain. So counter stain is the last stain which is used in this staining technique. It is the counter stain, that is safranin or basic fuchsin. And for the staining or for this gram staining, it is you need a bacterial culture for the preparation of the smear. Then we need all the requirement, that is biolope, it is required, bacterial culture, it is required, microscope, it is required, glass slides, they are also required. So all these requirements must be with you, and then we have to prepare the bacterial smear. So a slide is taken, then at the center of the slide a marking area is made onto the center of the slide with the glass marking pencil. And then we have to label the slide with the name with the initial of the organism, and then we have to take a loopful culture and we have to place into the center of the slide and we have to make a bacterial smear. So already in the previous video we have seen the preparation of bacterial smear. So now we are going to talk about the principle of the gram staining. What is the principle of the gram staining? So as I told, the gram staining, it is based on the cell wall composition of bacteria. The cell wall of the gram positive bacteria, it is thick, it is containing more peptidoglycan, it is containing less lipid, the cell wall of the gram-negative bacteria, it is thin and it is complex. It is containing less peptidoglycan, it is containing more lipid.